One thing that we do have for our array data, which I would like to get for our chip, is they looked at early generation of both the control and the bevacizumab treated, and then multiple generations of the bevacizumab treated, and we can compare expression across all of those combinations. So the idea is that I could hopefully do that as well, just knowing how much time I've had.
Are you seeing fewer patients ever being treated with bevacizumab? Given the data you showed, I was wondering if clinically people are changing their practices and are not necessarily giving it, given the toxicity, given the curve shifts. I mean, if I were a patient, I'm not sure I would get presented with the data. I'm not sure I would want to take it. And so I was just curious if there has been any sort of change in the clinical practice.
That's a really great question. I'll have to go and ask my mentor. I know him specifically that he still does use it, but that it's not with as much enthusiasm, I guess, as previously. But I'm not sure about here at UCSF kind of what the.
I mean, the data obviously will still be valuable because you're getting important information about, whether it's better than this or some other drug that comes along, and the same thing could happen. So I think it's still valid information that would glean, even if practices do change with that particular drug.
And then I know nowadays we have like this whole complete sequencing kind of stuff where you just look at the entire genome expression, all the proteomics and genomics, and then kind of go from there to try to link to elevate stuff rather than sort of trying to target specifically which genes are activated and which ones aren't. Is that, like, I know that's kind of widely accepted in the breast cancer world already, but in brain cancer, is that something that's on the frontier, or where is that in the field relative to these more traditional techniques?
So there is a lab over at Mission Bay that's currently looking at that. They're looking at exome sequencing actually right now as a mechanism. It's still pretty expensive to do whole genome sequencing. At least I guess in the clinical setting. So I know that they're looking at that, but that's all I know of here.
I'm kind of curious why you chose to do this sort of analysis where your genes are being expressed and not leaving RNAs.
Yeah, so this mechanism I was actually we spent a long time talking about is this necessary if you have RNA array data and everything, and what I've really found is that in order to understand you can get a lot of expression data that way, but knowing what is happening more on an epigenetic level, you need to do something more like this where you're looking at certain histone modifications in order to get that exact. Acknowledgements, my mentor and all the Augie lab members. This is, we jokingly call this our flag. One of the undergrads in our lab over the summer drew all of us in the lab. It's very nice of her when she left. My two committee members and the CTRFP program and the Department of Neuroscience.